I am excited to introduce today's speaker. Courtney Shearer is a fourth-year PhD student in Deborah Marks' group at Harvard Medical School. She's interested in developing machine learning models to learn from past evolution, present population, and future experimental biology sequence data for therapeutic sequence design and predicting clinical variant effects. She's going to share with us some exciting work today. So please, if you have any questions, feel free to come up to the microphone or raise your hand. If you're online, feel free to raise your hand there or unmute to ask your question, or just type it in the chat. So thank you again, Courtney, for joining us.
Can you hear me?
Great. Okay, so the actual title of my talk is Can We Computationally Predict Variant Effects So We Can Do Fewer Experiments? And like was just said, feel free to come up and ask questions like as you like. You don't have to wait till the end. You also can wait till the end, like whatever you like. So this is like, it's more fun if you talk and ask questions, because the primers are a little bit more of an introduction. So the bulk of this talk in the beginning will be truly like, basics of like variant effect prediction. And then towards the middle half of the talk, I'll go into a couple specific modeling methodologies that the Marks Lab has put together over the years. So the beginning will feel hopefully not too intimidating, and certainly nice to listen to at 8:30 a.m. in the morning. So with all that, the main question we're answering here, not to make this overcomplicated, is can we computationally predict variant effects so we can do fewer experiments? Maybe. Okay, so what's the goal? The goal here is to learn what variants cause disease. In this simple picture I have here, we have a DNA strand on the left, and then an individual who has some non-desirable phenotype as depicted by red spots and a frowning demeanor. So how do these variants cause disease? That's a question we have. We always want to know, if we do see variants in an individual, how are they causing this non-desirable or potentially desirable, but in this case, non-desirable phenotype? Things we can use to try to assess this, we classically have used things like genomics, epigenetics, transcriptomics, proteomics, and then with all these things, we try to understand how these molecules might be manifesting and mechanisms and pathways that ultimately lead to the phenotypes we're interested in. So how do we think about this variation generally when it comes to, I didn't make this, I'm not like an artist, this is AI generated. So classically we'll think about things from like a proteome perspective, where you have tons of molecules being made, I suppose, from like, I guess this doesn't show the magnitude of the difference in distributions of the size of the genome that might produce these things. But the proteome will produce, proteins, and the non-coding genome usually won't produce proteins, as we try to define our annotations. But to get into the types of variation we might see, we'll start with a simple depiction here, where at the top we have some reference. And I mean, I'm sure we've all heard of single nucleotide variants, which are just single base pair changes. They occur approximately in 1 in every kilobase of sequencing data. We can also see things like small insertions and deletions, which are a bit more rare. And then we'll even start to see like predicted regions for structural and copy number variants, which are supposedly more rare, unless our sequencing data isn't good at picking them up. So anyway, the types of these single nucleotide variants that occur in proteins, they have a propensity to disrupt codons that will actually, be translated and turned into the protein structures. Of course, we can have variants that are silent, having no effect on the protein sequence, allegedly. And we can have missense variants, which result in an amino acid substitution. And then we can have nonsense variants that substitute a stop codon for an amino acid. So all of this is a little bit of a refresher. Keep with me. It'll maybe get more exciting for some of you. In addition to the proteome, I'm not going to go into any numerical descriptions here, but just invite this, complex interaction system that we might view when we're thinking about non-coding variants. And just as a speculation, this complex architecture is certain to invite disruptions by all types of variants of certain sizes, but as a foreshadowing to the future of what I'll talk about at the end, potentially longer variants might be more disruptive than single nucleotide variants. So how are variants distinguished? Well, a lot of times in population genetics literature, these are some ways that we like to distinguish how we talk about variants aside from where they are in the genome, aside from how big they are. We talk about them as reference versus alternate. We talk about them as with respect to their frequency in a population of interest. We might call them the major variant or the minor variant. We might talk about them in terms of which variant matches the most recent ancestor shared by a recent species. And with respect to humans, this might be primates or chimps, when we might call this ancestral versus derived. And we also talk about these variants in terms of their association with increased disease risk. So we might call these risk versus non-risk. Clever. Classifying variants. We can also classify these variants further by minor allele frequencies. So instead of just thinking about things as minor and major, you can start to think about the frequencies of these variants and maybe a spectrum like this, where on the far end of the spectrum, you have a variant occurring in one person. And then on the bottom side of this arrow, you can think of these in terms of strict cutoffs based on percentages. But we also use some of these words at the top, such as very rare, rare, low, and common. Additionally, we can expand this on the left a bit, and people will talk about variants using language such as de novo, if a variant is even part of one person being in somatic cells as opposed to germline. And these are just a few ways we can begin to describe variants when we're talking about them in literature. So how do we associate these variants with diseases? Because remember, that's our big question. That's what we want to know. We can describe these variants in many ways, but we're really concerned about this question. You can tell me all the ways you want to describe a variant. I don't care. I want to know, how does the variant cause the disease? So the bulk of genetics and statistical genetics has kind of looked at this kind of lens of association, where we begin to look at variants again on the bottom here in terms of their minor allele frequency, but we also want to know about their effect size, how much an individual variant actually contributes to the phenotype we're interested in. And we kind of classify variants into these broad, discrete regions of this two-dimensional coordinate system. And we can say what studies are used to find these causal variants. We can have variants that are extremely unlikely. They're common variants with major effects. It's extremely unlikely to find those. We have GWAS studies, which are very good, allegedly, at finding low-frequency variants with intermediate effects, or common variants with small effects. We have rare variant analysis, which is good at finding rare variants with small effect size. And then we can use some things like tools like family studies to look for rare variants with major effects, and even sometimes grab some of those low frequency variants with intermediate effects. So all that is great, finding these associations, but even if we find them, there still is this big question here of, can we predict how the variants affect the trait? So what about all this stuff in the middle? How are we going to get there? So we could do this experimentally. We can use tools to look at the proteome, such as deep mutational scans, where we basically perturb each amino acid and see how it changes something simple like the protein folding, or some downstream assay that we're interested in. We can also look at more so, I guess, as this is called for the non-coding regions, we can look at what are called MPRAs, massively parallel reporter assays, where we essentially exogenously go in and stuff our variant of interest into a plasmid and see if we have potentially expression happening from some GFP, et cetera, or we can even assess things like star-seq for looking at enhancer binding, et cetera. We also can look at things like CRISPR mutagenesis, where we can potentially go in and directly, edit regions of the endogenous cell and see how those edits, manifest in some downstream cellular phenotype, and even further look at those at the organism level, but that seems fairly expensive. So why not do all these experiments? Seems like a good idea. Well, the human genome has about 20,000 protein coding genes. Average protein length is 470 amino acids. Possible amino acid substitutions per position is approximately 19. Variance per protein is around that number. This leads to a very large total of possible amino acid substitutions, just singles. For double mutants, this goes up to 15.9 trillion combinations, and triple mutations becomes astronomical. Okay, well, I don't care. I'm going to do a low throughput experiment anyway. Okay, so a cloning and DNA preparation might take 2 days. Protein expression, 2 to 3 days. Functional assays, 1 to 3 days. Data analysis, 1 day, not so bad. Total time for variants, 7 days. You have a team of 5 researchers. You're going to test your variants in parallel 50 per week. Okay, it's going to take you about 68,692 years. Okay, I'm going to do a high throughput experiment. Low throughput experiments, that's old news. Okay, I'm going to do 50,000 variants per experiment. That's going to take me about a month. And then I'm going to do this. That's still going to take me about 300 years. Ah, dang. Okay, so I should never do an experiment again. No. You can use prediction from some magical oracle tools, which I'll try to convince you about next, for this initial screening of variants you might want to test. Then you can validate these high impact variants experimentally. And then you can do this in a continuous feedback loop to improve predictions. Yes.
One thing I always think about with your previous slide is, are you testing it even in the right cell type?
True.
And so do you need to multiply all of those by all of the different cell types and states that ever exist in the human body?
Yeah, and we only define cell types that we define discrete cell types, but of course, that's a bit artificial. So it's essentially infinite if you wanted to time point any potential cell as it's differentiating. Okay, so maybe you believe me. Okay, so anyway, so this is a body of work from a group of us, the Variant Effect Prediction Association, led by Joe Marsh. We put out this guidelines for releasing a variant effect predictor. You can check it out if you like. It has some good descriptions of what a variant effect predictor would do. But essentially, a variant effect predictor, it aims to take in data, information from maybe these potential modalities on the left, and be useful, for highlighting variants with this evidence that you can use to filter down the variants you might want to be testing to see if they have clinical relevance. So what types of models are there? What types of variant effect predictors are there? How can we think about this? Well, we can have supervised models. We can have unsupervised models. We can have ensemble models. Supervised models, they're great. We can learn directly from known disease variants, and we get strong performance on well-studied variant types. Requires labels. If you have it, use it. Unsupervised models, main advantage, no reliance on disease labels, better generalization to unseen variants. Maybe we can use these to find things we don't already know about. And we can utilize evolutionary sequence data. In theory, you can use other things, but in the context of the variant effect prediction models that are out there, a lot of them you'll see using evolutionary sequence data right now. You can also do an ensemble model. You might have heard CAD, things like this, where they aim to combine multiple sources of evidence together, which can be very powerful depending on your question. For the sake of this talk, we're going to continue and we're going to focus on unsupervised models. because this seems to be the next frontier in terms of like being able to potentially say things about variants we don't know things about. Going into this space of better generalization to unseen variants. Unseen variants, I think we've seen all the variants. No way. So why unsupervised models are game changing? So this is just a simple analysis looking at, I believe, ClinGen and Nomad. What we have, this is just focusing on the 3% of the genome that is proteome. So you can imagine, like, if you had a bigger pie chart, like how much we wouldn't know about the non-coding genome. But this is kind of the labeled data we have. So it's pretty awesome to think about the potential for some of these other methods in terms of providing us evidence for going in and, learning things about these variants that we've never seen or been able to characterize before. So now we'll go into the part of the talk where I try to sell you some of the models, that we work on in my lab, in the Mark's lab. So these models are particularly interesting because they rely on this assumption that we can use this piece of orthogonal information for human genetics that's found in the 8.7 million species in the biosphere. So we can look at evolutionary sequence data where, in principle, this evolution has done a ton of experiments for us already to highlight variants that are desirable and to tell us stories about variants that maybe weren't desirable. And so if we see those variants of either category in the human population, maybe we can learn a bit of evidence as to what variants might be more or less desirable. So the distribution of sequences across all these organisms teaches us a lot about constraint. Like I just said, here's like a mathematical formulation of what I just said. We can think about potentially variants that are less probable, meaning they're less likely to be seen in this like learned distribution of sequences we see in evolution. They're less probable, they're probably less fit, and we can create that as like kind of a proxy for deleteriousness, a proxy. This is not 100% this is deleterious, this is evidence. Okay, so to go into this a little bit more, deep learning models capture complex conservation patterns, which take the full sequence context into account. So looking for patterns in sequence constraint is a relatively old idea. Until recently, only column conservation was considered. In this model, you say, oh, I'm going to take all my sequences, I'm going to line them up, and I'm going to say, oh, which one happened the most often, which one didn't happen the most often. That's a very simple idea. If only biology was that simple. As like a next step on top of that idea, we might start to look at pairs. So this might be best described in proteins when you're seeing pairs because of contacts, where if you see residues in contact with each other, presumably if one has a disruption, you're going to see a disruption in the other one. So this is a somewhat, I'm going to say old, but like not breaking news. Oh, that's nice. Okay. So some other potential models you might consider, you might ask yourself, oh, well, what about beyond pairs? What if there's more interesting higher dimensional interactions? Well, things like variational autoencoders, other types of models, they start to address this question of like going into this potentially nonlinear space mathematically and being able to pull out some of these higher order dependencies. We also see a lot of models these days. You'll see from like protein language models, et cetera, which also use slightly different strategies. A lot of times they don't necessarily rely on strict alignments of your data, and that can be really advantageous, especially for proteins if they don't align very well, such as antibodies, which might have hypervariable regions, et cetera. And then you can imagine how this is even more attractive potentially, which I'll get to towards the end, if you wanted to look at non-coding aspects of the genome where you have less constraint defined by structure that you would see in proteins, and they become less amenable to alignment-based models. So how do our models compare with lab experiments? Model predictions to lab experiments, yeah. Okay, so Eve predicts, Eve is one of our models. It's this model here, the VAE. So you can think of it as like a deep learning model that's able to pick up on higher order dependencies in the data. So just breaking beyond pairwise dependencies. It's still going to get pairwise. It's still going to get site-wise, but it can do more. And what we published this work back, geez, like 2021, I think now. So that was like right when I started my PhD. And what we're seeing here is that basically the model is able to predict, you know, if a variant is benign or pathogenic, just as well as if you took some deep mutational scanning experiment and put that up to the same task. So it does, it's as well as the experiment. So in theory, if you never had, this model's not trained on the experimental data. We made the experiment to confirm this or to find new things. So in theory, you never needed to do the experiment because we don't do experiments to validate our machine learning models. We do them to, well, we might do that, but we should be doing them to find evidence for clinical variants. So Eve is good at predicting clinical labels, but many variant effect predictors perform well at predicting clinical labels and correlate well with experimental data. But they are not designed for comparing variants in distinct genes. We're here on the right. Originally, we have an Eve model here that is unable to distinguish its scores for two different classes of genes. The purple one, or the one on the top, being haploinsufficient genes, where you would expect a disruption in that gene to be quite deleterious, whereas compared to a loss of function tolerated gene, where if you had a variant in that gene, you wouldn't expect it to be quite as deleterious as the haploinsufficient gene. So what we did is we said, oh, well, what if we could combine this evolutionary information with information from biobanks? So we use a two-step modular approach for building a whole proteome model. Where in this example, we're not taking like a single Eve model for a single protein. We're going to make models for the whole proteome. I don't think we've quite reached 20,000, but we've got a lot. And with this model, we basically have a variable for an evolutionary score that we're getting from these Eve models or other potential protein language models that might be ensembled in some way. And we basically do a conditional distribution where we're taking the probability of a variant being seen in some biobank, in this case, the UK biobank, and we're conditioning this on its evolutionary score. And we can parameterize this as a simple Bernoulli distribution, where our prior is Gaussian process prior. And aside from this Gaussian process prior, what's cool about this is we also can have some measure of uncertainty in the model. So sometimes the model will just give you a score and they'll tell you how to interpret that score and you're like, great, but wouldn't it be nice to know how uncertain the model is about that score? Because that might give you an additional piece of evidence as to say, like, how much can I trust this? So parameterizing models like this can be a powerful technique for giving the researcher more, more information about what the model is able to discern. So in this case, we see on the top with our Eve model, our original Eve model, which had no population data, and it's a little bit confused about how to score the haploinsufficient genes versus the loss of function tolerant genes. And then we see once we do train a new model with PopEve, we see, ignore the actual numerics on the x-axis there. It's about the comparison of the two box plots, not the actual numbers. So what we're seeing here is that the blue box plot is slightly shifted higher, meaning it has a higher likelihood of happening, meaning it's less deleterious, versus the purple box, it's shifted left, meaning it has a lower likelihood of being seen under the distribution, and therefore it is predicted to be more deleterious. Yes? Which Eve or PopEve?
The Eve model, yeah. Why can't it distinguish between the red and the green ones? Presumably like haploinsufficient genes would have maybe more like sequence
conservation than LOF tolerant genes
Yeah,
across species?
I think it would, but it's, you know, I think like the hypothesis here is that, you know, you're losing some of those like characteristics are based on what we've seen in humans. And you might want to have more current standing variation considered heavily in your model, which is why we add the additional information from human populations to kind of get the model to say, okay, I'm going to focus more on the human variation that I'm seeing, but also consider some of this evolutionary sequence information as a prior.
That makes sense.
And this is just like, you can see it in the data, like that's true, but like that's the intuition. Yeah, yeah. Okay. So you can try out these tools. On the left is Eve, and I guess there's no links here for you, but you can Google them for trying out Eve and PopEve mutation portals. They're pretty cool. They'll also, I don't know if it's Eve or PopEve, they'll also let you see the mutation like in a 3-dimensional structure. This is a little bit like shortchanging them, just showing you this like ugly like front door. But like, well, it's not ugly, but it's like not as cool as it could be if you put it in an actual thing to search. They're cooler than that, I promise you. Okay. So how can we evaluate these models? So now I'm getting towards the end, but this is a very cool piece of work that Hilary Finucane and others here at the Broad have worked on, where they've basically made this collection of benchmarks, and they call them genetics gym, where, you know, there's a lot of variant effect predictors out there. Which one do I use for my problem, for my variance? Who knows? I don't really believe in this world where one model is king. I think based on your question and based on the training data of the model, you should decide, you know, which piece of evidence to consider. I personally don't believe in this like God model that you can just feed all of this data into and it's going to give you some like crazy answer. Okay, so how can we evaluate these models? In this paper, I believe they looked at PopEve, which was the model I just showed you. This model called Misfit. Sorry if I don't know all of these models. MPC, they looked at alpha missense, which I believe we had a talk about earlier in the year. ESM1V, primate AI3D. And the first, wish that was a little bigger, sorry about that. In the first evaluation, they basically looked at de novo mutations in severe disorders, where they're comparing mutations in individuals with developmental disorders, et cetera, versus unaffected controls. And for this particular case, you see models like PopEve really shine, which is awesome. You know, we should just go home. We solved everything. Let's not look at anything else. We're the winners. Clap, please clap. Okay, no. Okay, so now we're going to look at some other evaluations because, you know, this is science and we need to not basically champion our own methods. We need to say, okay, what methods globally would be good for different questions. So for this next question, they're looking at rare variants in psychiatric conditions where you can kind of, I didn't mention this, sorry, but you can kind of think as I go across here, you can kind of think of this as a spectrum. Whereas on the left, you might have things that are under the highest constraints, such as de novo variants. So if you recall back to my beginning of the presentation when I had that spectrum, so the ones all the way on the left would be like variants that are under the strongest selection. They're not going to occur very often. So PopEve is very good at picking those up. But for the next one, we might think of these as being under moderate selection, potentially. We see other models potentially really shining at this. And I suppose you could read the paper to get more of an expose as to why some of these underlying training data and model architectures are particularly well suited for picking up signal from these different variant classes, called genetics gym. So in this next one, they're looking at common disease variants, which are under weaker selection. And they're looking at rare and common variants associated with various phenotypes in different biobank data, like UK Biobank, NGen, etc. And in this last one, they're looking at fine map variants. So they're looking at population genetics validation data using newly observed variants and the new NOMAD v4 data, but not in earlier versions of NOMAD, to independently assess both gene level and variant level prediction of performance. Maybe I should have had that at the beginning. But anyway, so that's the selection pressure. So what you can see here is these tasks, you can kind of conceptually think about them as being more or less, having more or less selection pressure. And we can see very nicely here that different models, some of them look like rock stars in the beginning. Some of them look like rock stars at the end. It's really up to us, like as computationalists who want to use these tools, build these tools, to think critically about what the underlying data is and not blindly jump on the hype train of new model. I wonder if this is going to give me the answer. Like, no, consider them all. Think about it. Look for really well done guideline studies such as this to kind of give you some bearings of how to like interact with the models in this field. So this is something I'm working on in my PhD. Don't read all this. Sorry, I should have done an animation. But basically, what you can now say, okay, what about unsupervised non-coding variant effect prediction? I talked a lot about proteins. What about non-coding variant effect prediction? So this is like a big mess. But people are trying to get at this question. We've seen like this year, small conference papers such as BEND, which one of my collaborators wrote, where they basically took all some of these different models you see here on the right that are allegedly training on non-coding information in an unsupervised way, and they're subjecting them to a lot of different tasks. But I highlighted in the red box here this variant effects task, and there is still a lot of room for improvement on this task, where the variant effect here is going from that beginning DNA sequence all the way to the person with the red spots on their face. So a lot of these tasks also here on the left, you can think of them as like, oh, can I go from sequence to RNA-seq expression? Can I go from sequence to epigenetics? Like those are good questions, but you have to keep in mind, we don't know always, like when we have a change in expression, if that's good or if that's bad. It just is. So there's a lot of room here for being able to go all the way across from molecular genomic information to phenotypic information. And you can check out, I just put a preprint out earlier, I don't know, I think last week, on my new model called LALIB, which is basically trying to do this, where essentially it trains on promoter regions explicitly with across evolution in tandem with protein language model embeddings to give you a prediction of the deleterious or pathogenic effect of indels. And that's not all I'll talk about here because it's kind of in the weeds, but you can check it out or ask me about it afterwards. But not saying this is solving the problem at all, but it is like trying to get at that question again of if we can do, find mutations in our genetics that go all the way to phenotype in the non-coding space. So you want to build a variant effect predictor too. How noble. So here is a commentary that we just put out from the Marks lab that you can read. I think it came out like 2 or 3 days ago on doing towards trustable use of machine learning models of variant effects in the clinic. And this is a very important piece of work to read because it's going to highlight some of these questions of like, you give it model data and you ask it to predict something, it will. But like, maybe you should ask the question of what is it training on? And then is this like relevant for what I'm interested in? You have to ask these questions or else you're blindly trusting something. And when it comes to clinical problems, we can blindly trust nothing. All right. So thanks for your time. Here's some pictures. Thanks to Harvard. I think Debbie's also associated with the Broad, so thanks to the Broad. And then thanks to the NSF for funding me. And yeah, I'll take any questions if anyone has any.
Thank you, Courtney, for an amazing presentation. Please, if anyone has a question, come on up to the microphone.
Do you think that models can eventually be able to understand interactions between variants? Because if a person has a deleterious variant, maybe they have a variant that compensates and then it doesn't affect them at all, that kind of thing.
Love that question. Can they do it now? I don't think so, but I think I would like to believe that given the right training paradigm, they could pick that out. But I think it's hard to have good validation data for those questions. So that's my answer. But I think that is an amazing question that I'm very interested in, because you could imagine like even some of these protein language models that are predicting clinical effects, even such as POP, alpha-missense, whatever, okay? They predict that this variant is potentially deleterious or pathogenic. Maybe it's not so bad if there's a compensating variant and a promoter that turns that gene completely off. So I don't know. I think that's very important for like getting like a global picture of variation in the individual. But I think that's the question I'm trying to get at maybe with my models by coupling promoter regions with the actual protein embeddings. Is then you could actually ask the model that question. Like I could ask the model that question right now today. Would I know if it's right? Well, I gotta find some good validation data. So that's a bit harder. Yeah.
I know that there have been many papers that have kind of set out when people are doing functional assays, what are the characteristics of those assays that make them most amenable to subsequent use in model building? Do you have any particular favorite traits for those of us who do design functional assays in terms of either how the experiments are designed or how they're reported that make them more usable for your models?
I guess from my particular lens on like non-coding variation, I'm always looking for like an endogenous editing because I want to see how that variant actually manifests in the functions of the cell. And I'm always looking for things that are more lended towards growth screens, where you see the cell type growth change over time, because I think that's a bit more similar to what we might imagine as like more of the organism level change. So for me, when I think about like going all the way across that spectrum, I want to see things that do that. And I'm not as, not that it's not useful, but I'm not as interested in like these like intermediate molecular phenotypes. Oh, another one.
What is the like compute cost of running your models?
Well, I mean, I can't like just tell you like you give Rose can give an estimate, sure. For Pop Eve, here's an estimate.
We
for
needed
you.
like 500 more Eve models within a short period of time. And I think that we spent $5,000 on like Google Cloud for that. But it can be upwards of like 20 to 30,000 if you're doing a lot of models, which is why we have some, you know, industry people like ran a bunch of Eve models for us and they paid for it.
Yeah,
But they did it wrong.
There's also, I don't know the answer to that because for my models, like at Harvard, we have like a really nice cluster they built. So I have no idea how much that costs, but it's free to me. But like I was training Lollev on like really beefy GPUs, like H100s. Yeah, I don't know who paid for it. Yeah, so maybe that's a Debbie question. But like, yeah. I like to think it's nominal compared to experimental costs, but maybe I'm naive.
Great. Well, thank you so much for such an excellent talk today. Please go ahead and grab a little more breakfast and then we'll reconvene at 9:30 down the hall for MPG.